The third source of genetic variation is random fertilization. So that single egg is fertilized by a single sperm, but it's in a very random manner. So when we consider independent assortment as well as random fertilization together, that is going to increase the possibilities even more. So there is 8.5 million possibilities for independent assortment, and there's also 8.5 possibilities for random fertilization. So this gives us an amazing 72 trillion possibility for the variation of the gamete. So these additional gametes are introduced by the crossover then, which would increase this number exponentially. So again, the odds that you are who you are is greater than 72 trillion when we add in the crossover in addition to the independent assortment. So in summary, the three different factors are the independent assortment, the crossover of the homologs, and also the random fertilization. When we consider the patterns of inheritance, we tend to try to oversimplify the phenotypes into just one single gene just for explanation, but in reality, it's way more complicated than this. So there are few phenotypes that are traced to just a single gene. So most traits in reality are determined by multiple alleles or interaction of several gene pairs. So one of these examples is blood pressure. There are many, many different genes on different chromosomes that all influence blood pressure. And those various gene pairs can interact. Some can be turned on or turned off at different stages. Environmental factors could also affect those, turn them on and turn them off. So it's much more complicated than the oversimplification that we're doing for this chapter. This slide is on dominant recessive inheritance. So you were introduced briefly to the Punnett square. So you're not going to have to do Punnett squares, but you will need to know the general percentages based on the genotypes of the parents. So you need to identify genotype versus phenotype and know what those are. So a Punnett square is a diagram that sort of predicts the possible gene combinations, and they're really, really simple. They are just a table, which we'll see in the next slide. So if we consider this example of a dominant allele, which would be the capital A, remember we tend to use capital letters for dominant alleles. For recessive alleles, we use lowercase letters, in this case a lowercase a for albinism. So the homozygous ones are the ones with two of the same capital letters or two of the same lowercase letters, and those would be homozygous. Therefore, the heterozygous always is a capital letter and a lowercase letter. So the probability of mating two heterozygous parents, that would be this genotype, capital A, lowercase a, is always going to be the results that we see here. So this is always going to be the results from two heterozygous parents. So it doesn't really matter what specific allele we are considering. 25 or 50, 25% are going to be homozygous dominant. 50% are going to be heterozygous, and 25% are going to be homozygous recessive. So we'll see that shown in the Punnett square here. You can see the gender basically is irrelevant here because these are not sex-linked traits. We'll talk about them later on, but there is the male, in this case, has a is heterozygous, so capital A and lowercase a. The female is also heterozygous, so capital A, lowercase a. When we cross them, we basically combine those letters. So for this gamete, there would be two capital A's. This gamete would have a capital A and a lowercase a. This one, the same, would be heterozygous, capital A and lowercase a. It doesn't matter whether you put the capital A first or second. And finally, this one is going to have two lowercase a's. So the person then would be recessive and have the albinism phenotype. So remember, the genotype is the letters, but the phenotype is what you physically can see. So the probability is always going to be the same percentages. If we consider another gene, and this gene we're going to consider, we saw earlier when we talked about chiasma, is going to be for hair colors. So we're going to consider a dominant and a recessive gene. The capital B is going to be for brown hair. The lowercase b is going to be for blonde hair, and it's going to be the same exact results that we see on the right side of your slide. So if the mother or the father, it doesn't really matter where you put whether you put the mother, father on the top or on the side, one is going to be heterozygous, the other is also heterozygous. When we cross them, we end up getting two capital B's, a capital B and a lowercase b, another capital B and lowercase b. Again, it doesn't matter if you put the lowercase b first or last. And then finally, two lowercase b's. So here again, you can see that 25% of the time there is going to, the genotype would be homozygous recessive. 25% of the time, the genotype would be homozygous dominant. And then 50% of the time, the genotype would be heterozygous. So the genotype is 25, 50, 25, but the phenotype is always going to be 75 to 25. And that's because 3 out of 4 of them are going to express brown hair, and 25%, 1 out of 4, is going to express blonde hair.